Hello everyone, now let us discuss about Good Manufacturing Practices Guide for Active Pharmaceutical Ingredients Part 8. This comes under Q7 Quality Guideline of ICH. Now we will discuss about storage and distribution. Coming to warehousing procedures, facilities should be available for the storage of all materials under appropriate conditions. Example, controlled temperatures, humidity whenever necessary. Record should be maintained of these conditions if they are critical for the maintenance of material characteristics. Unless there is an alternative system to prevent the unintentional or unauthorized use of quarantined, rejected, returned or recalled materials, separate storage area should be assigned for their temporary storage until the decision as to their future use has been taken. Until the decision as to their future use has been taken. Until then, they must be stored under temporary storage areas. Coming to distribution procedures, APIs and intermediates should only be released from distribution to third parties after they have been released by quality unit. They can be transferred under quarantine to another unit until the company's control, under the company's control when authorized by quality unit. And if appropriate, controls and documentations are in place. APIs and the intermediates should be transported in a manner that does not affect their quality. Special transport or storage conditions for an API or intermediate should be stated on the label. The manufacturer should ensure that contract acceptor for transportation of the API or intermediate knows and follows the appropriate transport and storage conditions. A system should be in place by which distribution of each batch of the intermediate or API can be readily determined to permit its recall. Now let us discuss about laboratory. Now coming to general controls, the independent quality unit should have its own disposal adequate laboratory facilities. And there should be documented procedures describing the sampling, testing, approval or rejection of materials, recording and storage of laboratory data. Laboratory records should also be maintained. All specifications, sampling plans and test procedures should be scientifically sound and appropriate to ensure that raw materials, intermediates or APIs and the labels and packaging materials, they conform to the established standards of quality and or purity. Specifications and test procedures should be considered with those included in the registration and filing. There can be specifications in addition to those in the registration or filing. Specifications, sampling plans and test procedures including the changes to them should be drafted by the appropriate organizational unit, revised, approved by the quality unit. Appropriate specifications should be established for APIs in accordance with accepted standards and consistent with the manufacturing process. The specification should include a control of the impurities, for example, organic impurities, inorganic impurities, residual solvents. If the API has a specification for microbial purity, appropriate actions limit for total microbial counts, objectionable organisms should be established and met. If the API has a specification for endotoxins, appropriate action limits should be established and met. Laboratory controls should be followed and documented at the time of performance. Any departures from the above described procedures should be documented and explained. Any out of specification result obtained should be investigated and documented according to the procedure. This procedure should require analysis of data, assessment of whether a significant problem exists, allocation of the task for corrective actions and conclusions. Any resampling and or retesting after out of specification results should be performed according to the documented procedure. Reagents and standard solutions should be prepared and labeled following written procedures. Use by date should be applied as appropriate for analytical reagents or standard solutions. Primary reference standard should be obtained as appropriate for the manufacture of APIs. The source of each primary reference standard should be documented. Record should be maintained at each primary reference standard storage and use in accordance with the supplier's recommendation. Primary reference standards obtained from an officially recognized source are normally used without testing if stored under the conditions consistent with the supplier's recommendation. When a primary reference standard is not available from an officially recognized source, an in-house primary standard should be established. Appropriate testing should be performed to establish fully the identity and purity of the primary reference standard. Appropriate documentation of this testing should be maintained. Secondary reference standards should also be appropriately prepared, identified, tested, approved, and stored. The suitability of each batch of secondary reference standard should be determined prior to first use by comparing against a primary reference standard. Each batch of secondary reference standard should be periodically requalified in accordance with a written protocol. Coming off testing. Let us discuss about testing of intermediates in APIs. For each batch of intermediate or API, appropriate laboratory tests should be conducted to determine the conformance of specification. An impurity profile describing the identified and unidentified impurities present in a typical batch produced by a specific controlled production process should be normally established for each API. The impurity profile should include the identity and some quantity analytical destination. Example, retention time, the range of each impurity observed, classification of the identified impurity, whether it is inorganic, organic or solvent, and the impurity profile is normally dependent upon the production process and origin of the API. Impurity profiles are normally not necessary for APIs from herbal or animal tissue origin. The impurity profile should be compared at appropriate intervals against the impurity profile in the regulatory submission or compared against historical data in order to detect changes to the API resulting from the modification in raw materials, equipment operation parameters, or the production process. Appropriate microbial tests should be conducted on each batch of intermediate and API when microbial quality is specified. Coming to validation of analytical procedures. Analytical methods should be validated unless the method employed is included in the relevant pharmacopoeia or other recognized standard reference. The suitability of all testing methods used should be nonetheless verified under actual conditions of use and documented. Methods should be validated to include consideration of characteristics included in the ICH guidelines on validation of analytical methods. The degree of analytical validation performed should reflect the purpose of the analysis and the stage of the API production process. Appropriate qualification of analytical equipment should be considered before starting validation of analytical methods. Complete record should be maintained of any modification of the validated analytical method. Such records include the reason for modification, appropriate data to verify that the modification produces results that are accurate and reliable as the established method. Coming to certificates of analysis. Authentic certificate of analysis should be issued for each batch of intermediate or API on request. Information on the name of the intermediate or API, including when appropriate, its grade, batch number, date of release, should be provided on the certificate of analysis. For intermediate or APIs with an expiry date, the expiry date should be provided on the label and certificate of analysis. For intermediates or APIs with retest date, the retest date should be indicated on the label and or certificate of analysis. The certificate should list each test performed in accordance with compendial or customer requirements, including the acceptance limit and the numerical results obtained, if the test results are numeric. Certificates should be dated and signed by authorized personnel of the quality unit and should show them the name, address and telephone number of the original manufacturer. Where the analysis has been carried out by a repacker or reprocessor, the certificate of analysis should show the name, address and telephone number of the repacker or reprocessor and reference to the name of original manufacturer. If new certificates are issued by or on behalf of repackers or reprocessors, agents or brokers, these certificates should show the name, address, telephone number of the laboratory that performed the analysis. They should also contain a reference to the name and address of the original manufacturer and to the original batch certificate, a copy of which should be attached. Coming to stability monitoring of API, a documented ongoing testing program should be designed to monitor the stability characteristics of API, and the results should be used to confirm appropriate storage conditions and retest or expiry dates. The test procedures used in the stability testing should be validated and be stability indicative. Stability samples should be stored in containers that stimulate the market container. Simulate it. That is, it is similar to the market container. For example, if the API is marketed in bags within filter drums, stability samplings can be packed in bags of the same material in small scale drums or similar or identical material composition to the market drums. Normally, the first three commercial production batches should be placed on the stability monitoring program to confirm the retest or expiry date. However, where data from previous studies show that API is expected to remain stable for at least 2 years, a fewer than 3 batches can be used. Thereafter, at least one batch per year of API manufactured, unless none is produced that year, should be added to the stability monitoring program and retested at least annually to confirm the stability. For APIs with short half-lives, retesting should be done more frequently. For example, for those biotechnological or biological or other APIs with shelf lives of 1 year or less, stability sampling should be obtained and should be tested monthly for the first 3 months and at 3 months interval after that. When data exists that confirm that the stability of the API is not compromised, elimination of specific test intervals, for example 9 months testing, can be considered. When appropriate, the stability storage condition should be considered with the ICH guidelines on stability. Coming to expiry and retest dating. When an intermediate is intended to be transferred outside the control of the manufacturer's material management system and an expiry or retest date is assigned, supporting stability information should be available. Example, published data and test results. An API expiry or retest should be based on the evaluation of data derived from stability studies. Common practices is to use a retest date, not an expiration date. Preliminary API expiry or retest date can be based on pilot scale batches if the pilot batches employ a method of manufacture and produce that simulates the final process to be used on commercial manufacturing scale, and the quality of the API represents the material to be made on a commercial scale. A representative sample should be taken for the purpose of performing a test. Reserve or retention samples. The packaging and holding of reserve samples is for the purpose of potential future evaluation of the quality of the batches of API and not for the future stability testing purposes. Appropriately identified reserve samples of each API batch should be retained for 1 year after the expiry date of the batch assigned by the manufacturer, or for 3 years after the distribution of the batch, whichever is longer. For APIs with retest date, similar reserve samples should be retained for 3 years after the batch is completely distributed by the manufacturer. The reserve sample should be stored in the same packaging system in which API is stored or in one that is equivalent to or more protective than the marketed packing system. Sufficient quantity should be retained to conduct at least 2 full compendial analysis, or there is no pharmacopoeial when there is no pharmacopoeial monograph, 2 full specifications analysis. Thank you for watching. Please subscribe for further videos on pharmaceutical sciences and other related disciplines.